In a sense, this is have a capacity of being an exocrine, and as well it is an endocrine organ. So before we go in much, we have to study the two of the aspects of this thing. One is that this particular gland has a exocrine property that we are going to identify, which is plenty or far more than that of endocrine characteristics. So this endocrine part is smaller, sparse, distributed, and it is present as a droplets. So we call them as islets of Langerhans, which is the endocrine capacity of this particular gland. So principally this particular gland, say for example 90 to 95% is having an exocrine capacity. So basically exocrine capacity with the seeds of endocrine islets which are placed. So if you see this particular gland, whenever you are seeing this particular anatomy of it, it has a head, then it has a constriction neck, then it had a body, then it is ready to tail. So this particular region, which is body, neck and this particular head, are not having that much of endocrine component. Only this particular tail component have got this particular like a sparse distributions of this particular element. We call them as islets of Langerhans. So these particular things, if you are making a sections over here in this region, only we are going to see these both of these entities. Whether you are making a cross section in the head region or neck region or body, we are principally seeing this particular exocrine capacities. So depending on where you are taking the tissues, this particular tissue or this particular microanatomy of this particular gland reflects accordingly. Now we are going to study this particular gland with the microanatomy. Principally tissues are taken to this particular tail end. So we are going to see this particular endocrine capacity, which is 95% to 90, more than that could be, and little bit like a sparse distributions of the islets of Langerhans. So before we move into this part, we have to understand this exocrine part, then we are going to identify the endocrine thing. So whenever you are going to identify this particular exocrine, that means it's going to produce some secretion and that is thrown out in a tunnel or in a kind of gutter system. So this exocrine capacities have got two basic things. One is secretory unit. That means the part which is producing this particular chemical substance, we call them as enzymes. And these enzymes, once they are produced from these factories, they need to be thrown out or exported out. And exported out through this particular system, we call them as ducts. So this particular thing have got two elements. We call them as secretory unit, also called as acini. And we have these ducts. We have small ducts which is meeting with the bigger ducts and then further even bigger ducts like this. So in this pancreas we are going to classify the ducts. We call them as intercalated ducts, interlobular ducts, and then we have this main pancreatic ducts like this. So it is smaller to bigger to super big ducts like this. So if you are just like seeing these ducts, whenever you will be seeing, they will be having some kind of caliber, which is smaller caliber, bigger caliber and huge big caliber like this. So this duct system are of different morphology. You can understand small, big, medium, etc., how these duct systems are there, and the secretory units. So to just like make these things very easy and small or comprehensive, it has a secretory unit, we call them as acini, and then we have this particular element which is thrown out into it, we call them as ducts, and these particular ducts are meeting together and getting like this. So this unit we call them as acini, and the cells which are present in this acini we call them as acinar cells. So they are producing this particular enzymes or things which is coming out of it. It has a nucleus just like which is there, and it has a secretory unit, we call them as zymogen granules, like they are kind of like a stored enzymes like this. Whenever they are present, they are liberated into this particular duct system and channeled through this duct to this exterior through this different duct system. Now this cells which is present in here, we call them as acinar cells. which are predominantly secreting cells, which are producing this particular juices which are present in it. Temporarily they are stored and then liberated as these things is coming out. Then we have this particular duct cell or lining cells on the ducts. They are cells which are cubical cells and have a tight junctions so that nothing escapes between them. So they are lining this and they are lining this particular elements as well, which are going to be the beaker ducts like intercalated to this particular interlobular to this main duct or main pancreatic duct. So we have two elements in this exocrine capacity. One, A, we call them as acini, which is holding this particular cells, acinar cells predominantly, and then we have this particular ducts, which is having three different entities, 1, 2, 3, small duct, large duct and intermediate ducts, which are going into it. Other than these acini cells, we have these other cells which is acinar helping cells. We are going to talk about little bit about acinar helping cells. They are cells which are basically lining between the intercalated cells and this particular acini or secretory unit. So we are just like going to learn about that, but I do not want you to make things complicated out at this point. So I want you to focus this secretory unit, which we call them as secretory unit of this particular exocrine entity. We call them as acini and the ducts. These are the two simple things. Acini are occupied by the cells which are producing the juices. We call them as acini cells. And then the channel system, the duct system. Duct system are small, large, and in between them we have the intermediate. Based on them we have different names to that. And what are the functions of this? Depending on the stimuli, this acini cell secretes some kind of chemical substance, we call them as enzymes. They are liberated through this particular duct system and channeled out and appropriately going to the part where it is required to be discharged. This is about this secretory unit, which is exocrine gland. Now, this particular gland, as we are seeing here, is having this kind of morphology, have a capsule covering to it. So if you see, this particular gland has a capsule which is a lining of this, and this particular capsules, if you see this, it breaks down into these particular glands, penetrating through here and breaking down in these particular glands into smaller lobes and smaller lobules. So whenever you are studying this particular glands microanatomically, you will see that this particular head, for example, is divided by several districts like this and smaller units. We call them as lobes, and even lobes are broken down into smaller districts. We call them as lobules. So within this we have these acini cells and the secretory unit and the duct system present. Along with this, rest of the unit are placed like this. So this particular pancreas tissue having a covering, we call them as septa, or we call this as capsule. The capsule penetrates deep into these particular tissues and splits into small lobes, and small lobes are fragmented even smaller into lobules. And if you take these lobules, we are going to see the secretory unit and the duct system which is present in it, which is the exocrine property. Now if you are taking this particular tissue from this tail end of the pancreas, we are going to see this exocrine component, but as well as we are going to see the endocrine component as well. So within this particular layers of this particular parenchymas, we are going to see sparse distributions of these particular collected regions. Now if you look at the colors of this particular pancreas tissue, it will be the exocrine property will be deeply stained, and within this particular deeply stained we will find some islands or localized collections of the cells which are not deep. We call them as pale staining areas. So these particular pale staining areas which is present in between this particular pancreatic exocrine tissues, we call them as islets of Langerhans. These islets of Langerhans are endocrine factories or endocrine bodies which are present. They are composed of different types of cells, A, B, D, etc., producing different kinds of hormones which we know physiologically what they are, insulin, glucagon, etc., But I am not going to talk about the physiological functions at this point, because we are not going to identify the cells or micro cells which are present in these particular islands. Rather, we are going to identify these particular collections of the tissue. Now, how we are going to identify this collective tissue is that we are going to identify this particular pancreatic tissue as secretory unit, which is plentifully seen And within this particular islets of within this particular exocrine tissues, we are going to see identify some pale staining island tissues which are present. And those pale staining island tissues we are going to mark as islets of Langerhans. They look different than that of rest of the secretory exocrine units. Alright. So, two things we are going to identify in this area. Apart from that, duct system, if it is present on the sections, we are going to identify. So, this is the basic understanding of the pancreas, how we are going to identify. The tissues we are going to see is a glandular tissue, that means it is composed of the glands which is capable of producing secretions. So, that particular glandular unit we call them as acini. So, this functional unit of this glands we call them as acini unit. Acini is composed of two things which is known as this acini cells or para-acini cells, which is helping this particular gland or acini cells to perform together, and we have a duct system. Duct may be small, medium, large, depending on the ducts you are seeing, we give different names for our better identifications. The cells of the acini may be circular or pyramidal in nature, whereas the cells which is lining these ducts are cuboidal. That means they are different size and shape which is present in it, and they are highly stained because they have this enzymes. Whenever these dyes are thrown it, this particular enzymes picks the dyes up and makes this particular thing looks beautifully colored. Compared to this islets of Langerhans, they don't have that kind of secretory element to catch into it. So, they appear to be having a pale appearance compared to the secretory unit or exocrine. So, deep stained area and a pale stained area of the islet cells which looks different than that of this tissues which is exocrine tissues that you are going to identify. Now, as we are going through this particular basic demonstration, then we are just like identify these elements with these pictures on them that clears the doubts whatever we have regard to this pancreatic tissues. So, this particular pancreatic tissues which is a glandular tissue, capability of this particular glands is of exocrine and endocrine. Exocrine is far more greater than that of endocrine. If you consider this particular volumes of the gland is concerned, I am saying like more than 95% is of this exocrine capacity, less than that we have this endocrine capacity. And again, endocrine is not present throughout this particular regions of the glands. It is principally predominantly seen in the tail end of the pancreas with these endocrine capacities. We call them as islets of Langerhans, which is this endocrine capacity. So, we started this liver tissue along with this region, and we are going to talk about these particular slides or element. We call them as pancreatic tissues, having this gross part which is familiar with us, and our microanatomy is concerned of two elements. It is an exocrine part that we are going to see, and in between the exocrine parts there are some regions or dockets of the endocrine components are there. We call them as islets of Langerhans. So, this particular thing has a capsule, this particular hole, this particular gland having a capsule. This capsule breaches this particular glandular tissues going deep into it, and as it is going deep into the substance of the gland, it brings this particular parenchyma into small lobes, and then it brings out into the small lobules. So, this particular thing is lobules if you are catching these things up, and we are going to see this exocrine and endocrine part. So, exocrine part we are composed of, we call them as acini, which is a functional and structural unit of any exocrine gland. We call them the acini as pancreatic acini, as it is present in the pancreatic gland. If it was present on the salivary acini, we call them as salivary acini, etc., etc. So, this particular secretory glands or secretory capacity of this particular pancreatic acini is watery. That is why we call them a serous kind of acini. So, right. And this particular acini have got two basic elements which are present to it. So, we are going to see this particular simple architecture of the glands. This is the secretory unit or the acini, composed of the cells which is present in here. We call them as acinar cells. They are producing the secretions, and whatever they are making out here need to be channeled out through this particular ducts or channel system. Through this duct they are coming out. This duct which is joining with this acini unit, we call them as intercalated duct. This intercalated duct of the adjacent one joins together, we call them as interlobular ducts, lobular ducts, and then main pancreatic duct, just like they are coming up, small to intermediate to the bigger one. The lining cells of this are different. You can see these particular cells are polyhedral, circular, oval, or like a kind of triangular in shape. Compared to this one, flat, cells, and these are cuboidal flat cells. In starting with this particular flat cuboidal cells, running this from here, it is more flatter, looks like squamous cells. This one becomes taking up cells of this particular element, we call them as this cuboidal cells. So this is the channel system or this collecting channel system which is present. Beside this acini cells and the lining cells of the duct, we have these cells which is joining or present in between this acini and this duct. We call them as centroacini cells. They are helping or bridging cells between the acini and this particular ducts. So they are like a kind of like making up the space which is present in between them. So they are basically present in between the acini cells and the channel cells or this particular duct cells there. We call them as centroacinar cells. They are a little bit of different shape than that of these flat cells. They have a little bit like a cytoplasm more compared to this one. But anyways, microscopically we are not going to identify because they are far super zoomed. So we are not going to see them, but it should be looking like that. So we should be knowing there are two prime cells, acini cells and the duct cell which is lining to it, and the duct channel system going to small, intermediate and big system like this. So going back to this particular main thing about this particular pancreatic tissues, it has a duct system of the pancreas. So depending on this particular caliber of the duct can be classified intercalated, interlobular and this accessory pancreatic duct depending on this particular ducts which is there, right? So these are the ducts which is present in this particular element. Other than this secretory unit, we call them as pancreatic acini, serous acini which is having this particular cells predominantly the containing the fluid which is watery in nature. So these particular cells are producing this particular substances which is kept in the cytoplasm packed by this particular packing material as a granules. We call them as zymogen granules which are present like this particular dots like this. If you these dots, zymogen granules, they are filled with the enzymes, filled with this particular capacity of the digestive things. And then these things are liberated into the duct system which are named accordingly as we have seen into it. So this particular pancreatic tissue, if you are looking like this, these are highly stained. As you seeing this particular element, these are element which is splitting this particular pancreas into small lobes or lobules. These are capsules which are splitting the glands into smaller districts. So lobules or lobes which are present in it, and between this particular lobules here we have this entire thing which is known as the pancreatic exocrine capacities or exocrine element. Now you see that compared to here we see the localized cells which look dissimilar than that of exocrine element. It is pale area and localized and small islets like they are present. So these elements are visibly different than that of exocrine cell. These are the endocrine cells. They are studied, fitted within this particular exocrine pancreatic tissues. We call them as islets of Langerhans. These islets of Langerhans plentifully seen as a islet cells as we are moving from the head to tail region. So more toward the spleen side, we are going to see this particular element, right? So this element, this is one, this is one another, this is another. They are pale region compared to this one, and this particular pale clustered cells which is present in here, looking like an island floating on the seas of this exocrine element. We call them as islets of Langerhans. So whenever histologically you are going to identify, we are going to find these gross areas of this exocrine capacity containing of this particular acini, which is the secretory cells which is present within this region, acini cells which is present, so acini. And then we have this particular ducts which is present in this particular regions like ducts over here. This is far bigger, we call them as interlobular ducts. And if you see this particular duct which is smaller one, this one circular, this is the smaller ducts which is present. They are present in the beginning of the system. We call them as intercalated ducts. So their ducts having a different caliber, different look over here based on how they are placed. So this particular element have got this exocrine capacity of the acini with these ducts and pancreatic tissues which is holding this light stained areas of the clustered clump cell. We call them as islets of Langerhans. This particular thing, if you see this element as this one, intercalated duct, they are zoomed like this. They should be looking like this. So if you zoom this element which is intercalated duct, they should be looking like this, having a circular lumen and this surrounding cell, simple cuboidal epithelium. They should be lining this regions. And this thing, if you zoom this, you are going to see the cluster cells having different kinds of cells. They are different morphological cells are there, which is seen in electron microscopes like A cell, B cell, D cell, etc., etc. So these are the elements. So if you zoom this part which is present in here, they should be looking like this. So this is the acini cells, and next to that we have this central acinar cells, and then we have these channels of the ducts which is visible here. We call them as intercalated duct system, right? Now this is a schematic picture. That's how it should be looking like ideally, but that need to be corroborated with this particular element, how these things should be looking like. So this is how we are going to see this particular the pancreatic tissue. This is all regions of the pancreatic tissue. So once you get your eyes settled down with this particular high regions of these resolutions, you have to identify these pale regions here. So pale districts over here, scattered over here in the seas of exocrine capacity, we call them as islets of Langerhans. This is the hallmark to identify this pancreatic tissue. So these exocrine tissues which is present as an ocean, and within these oceans we have these islands which is floating out here, we call them as islets of Langerhans. And then you might find some small ducts, intercalated duct like this, or bigger caliber ducts which is like a lobular ducts or main ducts like this. This is another small ducts which you can see here, which is not pointed here, this one, circular one and the lumen inside. So these are like things, and these white tissues or which you are seeing here separating between it, these are the septas which are basically separating this particular entire pancreatic tissue in the small those districts which call them as lobules. So these pale areas are islets of Langerhans, and this whole thing is exocrine capacity of the pancreas, right? So this is you are going to see whenever you are seeing this particular in 10x or low magnifications like this. So you have to settle your eyes with these all things. You have to identify these big things first, which is the glandular tissues. This glandular tissue, once you identify, then you try to identify if you see any islands of pale staining regions. If you identify them, the cells looks like different clump cells which is present in here, having a kind of nucleus which looks like this, and whole thing is circumscribed and separated from the rest of these islands, separated from this rest of the surrounding areas. So surrounding areas is deeply stained, this is pale stained, doesn't have much of capsules which is basically holding it. But if you just like a super stretch it, you can see this presence of thin layer capsules or microcapsules may be separating these things out, but you cannot see that. So this is what we are going to see through naked eyes. If you zoom this particular element, cells which looks like this, so which is island of Langerhans. So you can see this difference of this staining. This is deep stain, this is pale stain. So this is a pale visible thing and more strikingly visible in here. So you need to settle your eyes once you see this. Initially you will see that it's all ocean. Now later on, once these things are settled down, you can try and identify this particular area. Oh, this is little pale areas or circumscribed pale areas, which are basically the islands. So this is the hallmark. So exocrine element and presence of islands of endocrine elements, we call them as this, and presence of this duct system which are present, intercalated ducts or larger ducts which are presented. So whenever you're going to identify this, you are going to see. And in physiology, they'll ask a lot about questions on this. The different kind of cells which are present in this particular Langerhans cells or islets of Langerhans, and their functions and what they do actually in their life in terms of producing some kind of actions or functions of it. So this is about this pancreatic tissue. We have three different types of pancreatic tissue. Slides are there. So try to identify this exocrine first, then we identify the separating margin, we call them as septas, which is visible separating this particular pancreas into smaller regions, and then try to identify this particular the islets of Langerhans. Please do not move the slides because most of the slides I have fitted with this particular islands visible in it. So this particular islands of Langerhans are visible in three of them. So you settle your eyes with this exocrine part, then these pale areas will be visible to it. And again, they are not plentifully like you are seeing here. So there are maybe one or two sections in a per microscope field you may be identifying them. So they are not plentiful out there as you are seeing here in this region.